In the previous video, we have gone through, we can say, first and second part. First part was attachment and second part was fusion. So now we should go with third part, that is reverse transcriptase. Third part we have reverse transcriptase. So here you can see once the virus get entry into the cell, as in the last video we have discussed about the fusion. When this virus got entry into the host cell. So once the virus got entry into the host cell, its uncoating occurs. So here what will first will happen? First virus get entered, virus entered, and when the virus is entered, now what will happen? First we can say uncoating will occur. Uncoating will occur. How, as you can see, we can say the genome is present and we can say the covering of genome is capsid. So this means here capsid will be removed. Uncoating will occur. This means the protein part or capsid will be removed. Okay, so capsid is removed. So now what will happen? So yeah, when this capsid means this protein shell is degraded. Now how it will remove? This capsid shell or this protein shell is degraded. And this protein degrade is done by the enzyme which is protease. Okay, which is protease. Okay, so this is done means this protein is degraded. Now what will happen? As now remaining part, now what part is remaining? So the part is remaining which include basically its genome and enzymes are remaining. And enzymes will release out, genome and enzyme will release out. So that will be what? Which enzymes? Reverse transcriptase enzyme, protease enzyme, and integrase enzyme will be released. Okay, these enzymes will be released. So now as viral genome possesses what? As viral genome possesses basically two identical single stranded RNA. Viral genome possesses two identical single stranded RNA. Okay, so two identical single and stranded RNA. So here what will occur here? By the help of this enzyme reverse transcriptase, the RNA will convert into cDNA. RNA will convert into cDNA by the help of enzyme which is reverse transcriptase enzyme. So now here what? cDNA from cDNA, this means single stranded DNA, then again double stranded DNA is formed. Now this is also done by reverse transcriptase. So remember this reverse transcriptase help in both, like the main work of reverse transcriptase is not only to convert, we can say, RNA into DNA, but reverse transcriptase have ability to form second step of DNA by DNA. Here if I can write reverse transcriptase have ability to convert single stranded DNA into double stranded DNA. Okay, so these both steps are done by reverse transcriptase. Okay, so now if we can go with another, we can say part. Now fourth part we have integration, but before integration let me draw here the picture of this reverse transcriptase so it will be more clear to you guys. Okay, so as in the last we were having the fusion was done like this. This was the fusion was done, and here we have what? Here we have genome. Now this we can say genome will release here. This is, we can say, genome, and this is host cell. Host cell is here. Okay, host. This is the genome which come out, and from this what will form? As I told you, from this we can say DNA is formed, and from this DNA, single strand DNA, the double stranded DNA is formed. Okay, so this is how, if we have what, first we have the, we can say 5 and 2 3 end. As you all remember, the RNA, we can say strand is 5 and 2 3 end. So the DNA strand will be formed, that will be 3 and 2 5 end. Okay, DNA strand is formed, that is 3 and 2 5 end. This means cDNA is formed. Now here you can see RNA act as a cDNA. This means from RNA the cDNA is formed. Now here cDNA will act as the template of another DNA. The cDNA will act as the template of another DNA. This means 3 and 5 end. So this will be 5 and 2 3 end, because the 3 end is acting as a template. This means the 3 end is acting as the RNA template for the another DNA. Okay, so I hope this is clear to you guys. Now we can say, yep, HIV cannot direct the translation because it have to first go from the reverse transcription. It have to convert single stranded DNA into double stranded DNA. Okay, single stranded DNA into double stranded DNA. Okay, so I hope that it is clear to you guys. Now we are going with another step, which is integration. Okay, this is the fourth step, which is integration. Okay, so now let's see in this integration what will happen. So as we can say, now double stranded DNA is formed, like it is integrated in with the host chromosome. So what double stranded DNA is integrated with host chromosome. Host chromosome. Okay, so when this is integrated with the host chromosome, so this means this is directly transported to nucleus. Host nucleus transported. Host host nucleus. Okay, so when this is transported, now this we can say integration is done by the enzyme which is known as integrase. Done by enzyme integrase. Okay, so this is done. Now what will happen? Now as you can see, now this virus will remain inside the nucleus for several hours. We can say several years. This around 10 years virus will remain inside the nucleus. And then virus will remain inside the nucleus. That virus is known as prophage virus or provirus. Prophage virus or provirus, which will stay inside the nucleus. Okay, so now you can see this integration is done by the enzyme which we have discussed, integrase, and now it will remain inside the, we can say, nucleus. So this is also known as latent period, or we can say dominant period. Dominant or latent period. Okay, so I hope this is clear to you guys. If I can draw a diagram for you guys, so this is like this, and this was here, it was coming, and this is now, we can say, this is the host DNA, this is the host DNA, and here we have, we can say, virus DNA. Okay, so this is how integration is done. Now we have another step, which is basically what, which is transcription. Another step, we have transcription. So let's discuss here. Fifth step, transcription. Okay, so here we have transcription. So as DNA is integrated, now when this DNA is integrated, okay, so from this DNA, what will form? This from this DNA, mRNA will form, and from this mRNA, this will form by using what? By using the host enzyme. Now, which enzyme is used here? Here, enzyme is used as mRNA is formed. So, enzyme is used RNA polymerase. RNA polymerase. And we all know that HIV do not contain a RNA polymerase enzyme. However, this is what? This is host enzyme. So mRNA is formed on, and from this mRNA what will form, and from this mRNA it will form its own protein. From this mRNA what will be formed? Viral protein will be formed. Viral proteins are formed. Okay, so as we know that mRNA code larger molecules of viruses. Here we can say mRNA code what? We can say structural, we can say polyprotein. Polyprotein are coded. Okay, so from these polyprotein, it will basically convert into basically smaller molecules, or we can say smaller. And this is done by the enzyme which is protease. Protease. Okay. So now before going to the another step, now let me tell you here one more thing. Like here we have what? HIV. Yep. Here we have gene regulation. Gene regulation. This means here we have two type of gene, which is turn on gene, another is turn off gene. Okay, so here we can say HIV is present in, we can say, inside the nucleus, but HIV gene is what? HIV gene is turned off. Okay, so this is turned off till 10 years. After 10 years, it will be turned on, so it will do infection. So now let's move towards another part, which is basically, yep, another we have assembly. Assembly. So what will occur in this assembly? As we know, like the viral structural proteins are formed. So these proteins are formed. This will convert into smaller proteins. So as smaller proteins are formed, so now what will happen? So structural protein basically assembles. So now HIV will be mature, and this will, we can say, make its own, we can say, structure, like we can say genome, RNA, and its enzyme enclosed in the shell. Genome, here HIV contains genome, which is genome, RNA, and its enzymes which were present. Enzymes will make a shell, means these will be included in the shell. Okay, or you can say capsule. So I hope till that it is clear to you guys. So now let me tell you, we can say another step, which is budding. Okay, which is budding. Budding. So now what will happen in this step? As we can see, after assembly, the virus HIV virus is immature. So the HIV virus will move outside by the, we can say, help of exocytosis. So it will borrow the membrane from host. So that's why HIV is envelope virus membrane from host. Okay, so in this stage we can say a bud is formed. Okay, so bud is formed means this is totally enclosed. So HIV is released when this bud is formed. Now this bud is released by exocytosis. This bud is released by exocytosis. Now HIV will release what? HIV is released, and which is completely infectious virus. Now HIV will be released. Now HIV is what? HIV is released, and this is complete infectious particle. Complete infectious particle means this have ability to now infect the lymphocyte. This has ability to infect the lymphocyte. So I hope that it is clear to you guys. So now let me give you, we can say, a bit concept of infection. Last part we have infection. Okay, so now the HIV infect other lymphocytes and pass with some situations. Okay, in fact, we can say infect to other lymphocytes and cause, we can say, passes through situations. So this was all about this HIV, life cycle of HIV. So I hope this is clear to you guys. So in the next video, we're gonna discuss about diseases. With that, bye bye.